This is part 5 of the Chapter 2 lecture notes, and I'm just going to go ahead and warn you ahead of time. I'm going to go a little bit faster and try to finish up because um, 4 15-minute lectures is probably the maximum that you need to endure per chapter. So I'm going to try to finish up this time a little bit quicker. So the only information that's here, I've already said it actually, is that the substances that will dissolve in water are going to be ions or ionic compounds and polar molecules. But if they are nonpolar, then they are insoluble in water. And in the case where something is dissolved in water, water is the solvent, and then the compound that is dissolved is the solute. This is an example of how water dissolves an ionic compound like table salt. When the table salt, sodium chloride, mixes in with the water, the sodium and the chloride, the sodium and the chlorine atoms form ions, and the sodium ion is positive, so it's attracted to the negative oxygen end of the water, and the water kind of forms a sphere around the sodium and separates it from the chloride ions. And the chloride ions have that same sphere, but since they're negative, they're attracted to the positive hydrogen end of the water molecules. So again, water surrounds the chloride ion, forms a sphere of hydration around the chloride ion in the same way it does around the sodium, except that it's just the molecule flips around. And these properties of water, the hydrogen bonding and the polar covalent nature of water, also make it cohesive. And cohesion is the tendency of water molecules to stick together due to hydrogen bonding. And cohesion allows for the development of surface tension. We see surface tension when something floats on the surface of water, like if you drop a pin or a needle on the surface of water in a beaker, it'll float. And capillary action, which is what we see when we see this in the veins or the vascular tissue of plants, that water is drawn up a tree from the roots all the way to the top of to the very top branches and leaves, and that happens by capillary action. Um, and we see it also in a capillary tube. Sometimes they'll use a capillary tube at the doctor's office to, once they have a drop of blood, they'll put the capillary tube on the drop, and then the blood will just kind of be sucked up into the tube. And that property, capillary action, is caused by adhesion and cohesion. Adhesion is the attraction between water molecules and other molecules. So in this case, the attraction between the water molecules and the glass of the tubing. But it wouldn't be possible if the water molecules weren't also attracted to each other, um, which is the cohesion. So capillary action is primarily due to adhesion, and then both properties allow for insects like a water strider to float on the surface of water. Um, and this is also very similar to surface tension, but cohesion and adhesion allow for that water strider to float on the surface. The water molecules are cohesive, they're attracted to each other, so it's hard to break through the surface of the water unless it's very heavy, which that water strider is not. And the adhesion allows the water to stick to the water strider's legs, so that allows it to float on the water. Now we're going to move on and talk about solutions and their pH. And pH is a measurement of the acidity or basicity, which we don't use that word. We use the word alkalinity. Alkalinity is the word that refers to the basic, whenever a solution is basic versus acidic. So pH is measured by the concentration of hydrogen ions in a solution. So solutions with a high hydrogen ion concentration are acids, and they're going to have a low pH. And solutions with high hydroxide concentrations, that means they have low hydrogen ion concentration, are alkaline or basic, and that means they're going to have a high pH. And this is a table that shows the pH levels from 0 all the way up to 14. 0 is most acidic and 14 is most basic. All right, so gastric acid, which is stomach acid, has a pH of about 1. Urine has a pH of about 6. Blood has, not on here, but has a pH somewhere around here, about 7.4. And bleach has a pH of 13. Ammonia is 11. So ammonia and bleach and soap are all bases. And the mathematics of calculating the pH is actually a logarithm based calculation. We're not going to calculate pH in this class, but just so that you understand, that is the reason why whenever the solution has a higher hydrogen ion concentration, it has a lower pH. And a solution with a low hydrogen ion concentration has a higher pH. Acids have a pH of less than 7, and bases have a pH of greater than 7. This is the same chemical formula that I showed you in a previous slide, and it is a group of chemicals that play a role as a buffer in the human body. And a buffer is a chemical or group of chemicals that help to maintain the pH of body fluids within a narrow range. So what will happen is if the pH in your body is high, that means that you maybe are dehydrated or maybe you have vomited a lot, because that tends to make your pH a little bit too high. And what will happen then is that carbon dioxide will combine with water, form carbonic acid. That acid will break up and release hydrogen ions and lower the pH. If the pH is too low, the opposite will happen. If the pH is too low, bicarbonate ions and hydrogen ions in your body fluids will combine to form carbonic acid, and carbonic acid will break up into water and carbon dioxide. And that will, in other words, it will lower the hydrogen ion concentration and raise the pH. And then this section is on carbon. The reason we have to go over carbon is because in chapter 3, all of the chemicals that we're going to learn about, they're called macromolecules. In nutrition, we hear this a lot, that you need to get your macros. That's just like a new popular term to use for carbohydrates, proteins, and fats, and fiber. Fiber is not listed up here, but that's in nutrition. But it's very much related to the molecules that are found in living things, because what do we eat? We eat other living things. Or highly processed forms of other living things, but our basic diet comes from plant and animal material. So macromolecules are going to be proteins, carbohydrates, lipids, which are fats, and nucleic acids, which would be your genetic material or your DNA. And they are carbon-based. They contain a lot of carbon. So why is carbon so special? So we know that water is special. So why is carbon special for living things? It can form 4 covalent bonds. Not 1, 2, or 3, but 4 covalent bonds with different atoms. It is in group 14, and so it's able to do that. And that allows it to serve as the backbone for macromolecules. Each carbon has 4 electrons in its valence shell, and that means it can gain 4 electrons or form 4 covalent bonds to achieve its stability, to achieve the octet rule. Hydrocarbons are found in many of these macromolecules, and they are exactly what the name sounds like. They are carbon plus hydrogen. Methane gas is a hydrocarbon. Methane gas has 1 carbon and 4 hydrogen atoms covalently bonded to that carbon. Burning hydrocarbons releases energy. This is a model of methane. And then some other gases, ethane and ethene, have different numbers of carbon and hydrogen atoms. These are, by the way, these are aliphatic hydrocarbons. Yeah, I know that you may see that term in your questions. These are aliphatic hydrocarbons. And then there's aromatic hydrocarbons, which form rings. So the aliphatic hydrocarbons don't form rings, they form chains. Well, this one doesn't, of course, because there's only one of them. But the aromatic hydrocarbons, cyclopentane, cyclohexane, benzene, pyridine, these all are aromatic because they release an odor, and they form rings. Isomers are different forms of molecules. They have the same chemical formula, but different shapes. So structural isomers have different arrangements of atoms. Geometric isomers have a different arrangements of atoms around a double bond. See here and here. Notice the CH3, both of the CH3 groups are on the bottom in cis-butene, and they're opposite one another in trans-butene. And then enantiomers are molecules that share chemical formula and bonds but different 3D placement of atoms. They're mirror images of one another. Trans versus cis molecules, the trans conformation puts the carbons on opposite sides of the double bond, so the trans conformation tends to be a straight chain, and the cis configuration tends to be a bent chain. It's bent. So fatty acids that have a double bond are unsaturated, and those are going to be fatty acids that are found in oils. And fatty acids with no double bonds are saturated. And the unsaturated would look like this, and the saturated would look like this. Saturated fats are the solid fats that come from animal sources, and then the unsaturated fats are the liquid fats that come from plant sources. Here is an example of 2 enantiomers, D-alanine and L-alanine. They're mirror images of each other, and only the L forms of amino acids are used to make proteins. We will talk more about that in the next chapter. Amino acids are the building blocks for proteins. Now, I really have to go fast, but it doesn't matter that much because I don't really ask a lot of questions. I don't focus a lot on functional groups, but in a lot of these macromolecules that we will study in chapter 3, you're going to see some chemical groups like are listed in this table. A hydroxyl group is just an O bonded to an H, and the R in all of these examples, the R just means it can be any chemical or any group of chemicals. R just means variable. So you can study this table if you would like, so you can recognize some of these chemical groups. And then I think this is the last one. This is showing hydrogen bonding, helping to form the double helix structure of DNA. So another example of hydrogen bonding which does not involve water. It helps form the double helix structure of DNA.